Welcome to Module 1, our course introduction. There are 3 things I'd like you to keep in mind while you're learning about synthetic biology. The first is that we're developing a framework for new knowledge. We're probably going to be talking about a lot of things that you haven't spent much time thinking about. We're going to try and group these new ideas into topics, like DNA or proteins. That way you'll know what area to refer to when you stumble across problems in your own work. Second, it turns out that biology has a lot of moving parts and is very complicated, and we can only control small pieces of it. So we'll try and focus on what are the moving parts, what lets us put in the least amount of effort to get the biggest amount of return. And third, and most importantly, synthetic biology really needs people with a diverse set of skills and backgrounds. We're going to really look for opportunities for you to use your existing skills to improve synthetic biology. One last warning before we start. Biology is a very complicated science, and there's all types of cool things that happen in biology that break the basic rules of engineering. For example, look at these 2 species of whiptail lizards, which are native to the southwestern United States. The lizard on the far left and the far right are members of the same species. They're sexually dimorphic. It means that the females and the males don't look the same. They mate, and the female lays eggs. Now the species in the middle is only female. Females mate with each other and both lay eggs. Eggs which are almost exact copies of their mothers. Now, when biology can do things this wild, how can we predictably engineer these systems? We can't, but we do the best we can, and we try to stick to systems that have a long history of being engineerable. Just don't be surprised when you find things might work a little differently in real life. You have to reproduce. If we want to simplify a cell to the absolute simplest machine that we can think of a black box where you put in food and you get out more boxes. This is a really good starting point for our work because it lets us think of an engineerable system where we might not know what's going on, but we have things we can measure. We start with a black box where we put in food and we get more boxes. Now, with real living systems, food is a bit too generic of a term. Cells need many things in order to survive. They need water, they need building material to make the cell parts, and they need a source of energy to manipulate those parts into their finished form. And they don't just make more cells. They make the individual parts of the cell. They make the machines that allow those parts to be made. And lastly, they have waste as well. In addition to cellular construction, cells can recognize other types of inputs as well. For example, if a cell notices a pheromone, it might recognize that as an opportunity to reproduce. By being able to respond to environmental cues above and beyond the presence of food, a cell can increase its fitness and reproduce more effectively. For example, an organism might respond to a pheromone. A pheromone is a chemical that is absorbed by an organism and lets it know there's a good opportunity to reproduce. When the cell receives this signal, it might start to reconfigure itself to be prepared for reproduction. By responding to signals other than just food, a cell can have a more robust life cycle. In the laboratory, we also make use of chemical signals in order to get cells to respond. We also use other environmental controls, like changing the temperature, the pH, or the presence of light, to get cells to do things like move, change their shape, or change their activity level. It's important for cells to be able to respond to negative stimuli as well. For example, if a cell begins to starve, that would not be a good opportunity for reproduction. So, cells contain mechanisms to process these types of signals as well. For example, if a cell is starving or being eaten by an attacker, it might hibernate or exude a poison. When we think about these inputs and outputs of cellular biology, how does this relate to synthetic biology? I like to think of the old European folktale of Rumpelstiltskin, who had his own inputs and outputs. He tried to take straw and spin it into gold. Another way of saying that is, he tried to take cheap stuff and turn it into valuable stuff. Synthetic biology works the same way. We're using our cells to take cheap foods and turn them into valuable products. Another way to use synthetic biology is to apply the lessons from life to other types of problems. Let's think about the space station. Astronauts breathe out about 1 kilogram of CO2 per day. We could just throw that CO2 into space, but wouldn't it be better if we had another way to reuse that carbon? Let's think about what happens to that CO2. When the astronaut eats food, and that combines with the oxygen that he breathes from the air. They combine to produce the energy and building materials that he needs to live. CO2 is the waste product of that reaction. That food gets to the space station by rocket. But on Earth, the food is probably grown in a field. Plants absorb sunlight and CO2 from the atmosphere in a process known as photosynthesis to produce sugar. We've gone from CO2 in the air to CO2 in the air. We now know a process that lets us capture CO2 in the air and turn it into food. We might not want to send dirt and cabbages to the space station, but synthetic biology lets us shortcut that process. Maybe we can use algae or yeast to take the carbon dioxide from the air in the space station and make food using synthetic biology. We've talked about what this little black box can do, but what's really inside that little black box? That's going to be the topic of modules 2, 3, and 4. In module 2, we'll describe the cell as a chemical factory. In module 3, we'll learn what are the real molecules inside that factory and how does that factory actually work. In module 4, we'll learn that we can control that factory through synthetic biology. While you're taking the course, check out the jargon busters in the external resources. There's just going to be some vocabulary you have to know in synthetic biology, and we've tried to simplify the process by providing short definitions to some key terms. For example, you'll hear me talking a lot about the central dogma of biology. While it may seem scary, this is just the simple idea that DNA makes RNA, which makes protein. This simple rule helps us use DNA to control all the processes of the cell. Remember that synthetic biology is also looking for help from you. There's some modern challenges in synthetic biology that we really struggle with, and we're looking for fresh ideas from fresh minds to help us address these challenges. First, what's best for the cell isn't what's always best for the synthetic biologist. When we manipulate a cell, and all it wants to do is reproduce, we run into a problem where cells will ignore our instructions in order to do their own thing. Remember the story of Rumpelstiltskin? He wasn't happy to simply spin straw into gold. He also wanted the prince's firstborn son as part of the deal. Trying to reduce these heavy costs is a big challenge in synthetic biology. Another challenge in synthetic biology is that it's very difficult to reuse your pieces, and this manifests itself in two ways. First, the individual components of the cell have evolved over billions of years to have very specific functions. When we try and use those parts for new functions, it can be difficult to get them engineered just right. Second, the cell is a three-dimensional bag, with things bumping around and moving all the time. It's not like a circuit board, where reactions proceed in a row. Sometimes, when we engineer new systems in synthetic biology, they bump into parts that cause other problems. Trying to find ways to eliminate this crosstalk is quite difficult. Biology is also just a noisy and messy process. Cells require lots of components in order to function properly, and sometimes it's difficult to tell which components are part of your system and which ones are part of the background. Trying to decouple these processes is another major challenge. Synthetic biology can also be quite messy on a laboratory scale. There's a video in the external resources about an electric car. Go ahead and watch that video now. Welcome back. In that video, every single piece of technology had its own waste stream, but in the real world, we plug our computers into the wall and electricity comes through a wire. That electricity is produced at a central facility. This centralization allows us to have better waste processing. On the lab scale, logistics can be quite difficult, as each individual lab needs to manage its own waste streams, as well as be able to keep the cells alive. In some ways, a synthetic biology laboratory is closer to a zoo than to an office building. Finding ways to centralize resources or otherwise reduce waste would be very useful for synthetic biologists. What can we do to address these challenges? Well, most projects in synthetic biology fall into one of two categories. Either we're making cool tools to enable further research, or we're doing the research that uses these cool tools to make new solutions. The tool makers and the project developers need to work together closely in order to drive progress forward. Let's recap. We're going to be developing a framework for knowledge around synthetic biology by finding convenient groupings for each topic we're studying. We're going to find the moving pieces, finding exactly where we can put the least amount of effort to get the biggest amount of gain in an engineerable system. And we're going to be looking to you for solutions to synthetic biology problems using the skills you already have.